You ready?
Yes. I want to thank the organizers for inviting me. It's a very, very exciting meeting, and I have learned really a lot. Oops. Got it. Yeah, I got it. Okay, thank you. So I will switch the topic, and I will talk today about myocardial infarction, which is characterized by immediate inflammatory response, extensive cardiomyocyte death, and replacement fibrosis to prevent cardiac rupture. And replacement fibrosis occurs in response to cardiomyocyte apoptosis, necrosis, inflammation, and is characterized by activation of fibroblasts, which produce collagen type 1 and produce fibrous scar. And this is the representative images of the mouse model of myocardial infarction, and serious red, and also here staining for alpha smooth muscle actin, demonstrates development of fibrous scar in mice. Development of fibrous scar is very positive because it prevents the rupture. And it is mediated by the crosstalk of endothelial cells, cardiomyocytes, mast cells, lymphocyte, and of course myofibroblasts. Myofibroblasts develop from fibroblasts from the liver resident fibroblasts, but they produce collagen. So in response to injury, cell death, production of reactive oxygen species, angiotensin II, high glucose, altered pH, activation of TGF-beta, which is produced by monocytes and inflammatory cells, leads to activation of fibroblasts into myofibroblasts, production extracellular matrix fibrous scar. But then there is a next phase when these myofibroblasts have to inactivate. They have to stop producing collagen. And one of the mechanisms by which they inactivate is they undergo senescence and clearance by immune cells. So there are several predispositions to myocardial infarction. And one, of course, is genetics. And out of many, many genes which are critical for development, atherosclerosis is linked to two genetic locuses, and this is this locus, and these mutations actually have been linked to these 4 genes. And out of these 4 genes, ApoE, ApoB, and LDL receptor are critical in the pathogenesis of atherosclerosis. These genetic factors are critical, but they are predispositions, so there should be other environmental factors which allow these factors to be causing the disease. So what are the other factors? And the other factors are epigenetic factors and gene expression profile. And several new technologies have been evolving in the recent years, which allow us to look at the more specific factors which are not involve genetic mutations. So one of these techniques is ATAC-seq, and allows us to look at the unraveling of the genomic DNA and opening chromatin, and usually it's linked to transcription. However, RNA-seq is the technique which allows us to understand if these genes are either transcribed or suppressed. Finally, proteomics allows us to actually understand if the levels of RNA are translated into the proteins and how these proteins are upregulated or downregulated or stable. And this is one example that combined analysis of RNA-seq and ATAC-seq gives us identification of all the cells that I already mentioned to you, and we see that fibroblast population plays an important role in development of healing during the myocardial infarction. And so we can actually analyze combined data on cardiomyocytes. The big changes in gene expression profile and epigenetic profile are shown in cardiomyocytes, but again, so fibroblasts play an important role in development of this transient fibrous scar. So we are going to focus on fibroblasts, and this is the data from mouse models of myocardial infarction. In normal heart, there are fibroblasts, but they do not produce alpha smooth muscle actin or collagen and do not proliferate. But in response to injury and release of TGF-beta, there is an activation and proliferation of fibroblasts, and by day 4 or 7, they start produce collagen and regulate alpha smooth muscle actin. But then after day 10, formation of scar is accomplished. And so these fibroblasts, they have to persist in the cardiac wall for a certain period of time to make sure that there is no rupture. But then they have to be eliminated. And one of the ways to be inactive, so they stop producing collagen and they stop proliferation. So what happens next? In the phase of proliferation or healing. So, and here we're looking for epigenetic profile of this fibroblast. No really big changes in the uninjured, but if we look at the day 3 and day 7 during the healing process, formation of fibrous scar, fibroblasts change the epigenetic profile, unravel the genomic DNA sites for transcription very strongly. And after 2 weeks during resolution phase or recovery from fibrosis, we see that the epigenetic profile changes dramatically, and there is a upregulation of genes that we haven't seen in the normal fibroblast. So this is translated to the gene expression profile, and again, so healing phase is associated with proliferation, formation of the stress fibers, migration, and deposition of extracellular matrix proteins. And this ends by day 7. With week 2 and week 4, we see that there is a resolution phase, and production of extracellular matrix and proliferation is accomplished, and there is an upregulation of very interesting group of genes which are linked to the cartilage production. And why is it important? Because those myofibroblasts or fibroblasts which exhibit an impaired phenotype, they potentially can contribute to heart fibrosis and lead to heart failure. This is another example of looking at the spatial transcriptomics. These techniques allows us to look at the specific populations, its specific part of the heart. So the heart is sectioned, hybridized with different transcripts, and finally released, so we can identify in the scar of the heart different population of myofibroblasts or fibroblasts, and different colors reflect their different position in the cardiac wall. But also they have different emergence of production of collagen different during different days. Like for example, the first is the early intracardiac scar is 3 days after the myocardial infarction, then 4 days intermediate, and 7 days is the final fibrous scar formation. So what happens during the resolution? After 2 weeks of myocardial infarction, myofibroblasts either apoptose, so they have this programmed death and they're cleared by immune cells, or they senesce. So they have this cycle arrest and they just hibernate in this cardiac wall. 65% of the cells are just waiting for their cell fate. And what happens to them afterwards? So some of these senescent cells are called helper cells. They actually have a very positive role. They promote cardiomyocyte maturation, maintain tissue homeostasis. They're resistant to TGF-beta, and they're susceptible to apoptosis. Approximately 3 weeks after the myocardial infarction, these cells are cleared by NK cells. But there is another group of senescent cells which are detrimental, and they persist in the heart after 6 weeks. They induce senescence of cardiomyocytes. So cardiomyocytes cannot populate this area, they cannot proliferate. They promote inflammation and cardiac fibrosis. They respond to TGF-beta and actually can proliferate and again, produce fibrous scar. And they're resistant to apoptosis. So these cells are critical for development of heart failure. Why is it happening in some patients and it doesn't happen in other patients? So development of senescent, detrimental senescent phenotype is linked to the telomere shortening, specific epigenetic changes which are linked to upregulation of this methyltransferase, accumulation of DNA damage, oxidative stress, mitochondrial dysfunction, and very important, the upregulation of P53, P16, P21, the genes which are critical for inhibition of cell cycle, while the survival genes are actually upregulated. Senescent, detrimental senescent cells, they also secrete senescent-associated secretory phenotype, a bunch of cytokines which affect nearest cells. They can be recognized by expression of beta-gal. They prevent their own removal through protective senescent cell anti-apoptotic pathway, SCAP. They promote senescence of surrounding cells, I already said. They promote aging and cancer. So what is the function of senescent-associated phenotype? So senescent cells secrete IL-6, IL-8, ataxin, different chemokines, CCL5, so IL-6, which are known as very highly inflammatory cytokines. Very important that CCL2, which is secreted by senescent fibroblasts, can prevent immune clearance. It's a sort of like suppresses activation of T cell and K cells. A recently identified pathway, activation of GATA4 in senescent fibroblasts, leads to mitochondrial damage. Another pathway has been suggested that serine proteinase activates TGF-beta IGFBP7 pathway in cardiomyocytes. And the authors of this paper also suggest that IGBP7 can be a marker of dying and failing cardiomyocytes. So what can we do with this? How can we get rid of this detrimental senescent cells? And senolytics are the biologically active compounds that specifically can remove senescent cells. They can target one or more of these pro-survival proteins, can preventively induce apoptosis in senescent cells. Targeted removal of senescent cells through agents called senolytics was shown to enhance lifespan, alleviate systemic inflammation, improve organ function, and also mitigate characteristic age-related disorders. So it's the future, the drug of the future. And this is one of the examples when in the post-myocardial recovery phase, senescent fibroblasts release SASP, and these cytokines induce senescence of cardiomyocytes and also stimulate production of fibrous scar. But if these mice are treated with senolytics, then senescent cells are eliminated. There are no production of SASP and inflammatory cytokines, and no formation of fibrous scar. And the drug that has been tested in this particular study was navitoclax. Several generations of senolytics have been generated. As David said, that original drugs were designed by, driven by hypothesis. And so what was the hypothesis? That first of all, we need to remove or the block, the genes that prevent apoptosis, and also the kinases which are associated with the senescent phenotype. Several lysosomal proteins. We need to neutralize P53, induce block cardiac glycosides, and also like heat shock proteins which prevent apoptosis and provide survival signals also should be blocked. But the recent generation of senolytic treatments is based on the high throughput compound library screening, vaccines, and one of the potential ways to eliminate senescent cells is generate the CAR-T vaccines. Recently, 2 papers were published, one in Science, one in Nature, suggesting that there is a way to target senescent detrimental fibroblasts using a fibroblast-associated protein. There are 2 other examples which I would like to mention. Cellular therapies, cell stem therapies can be potentially useful for treatment of myocardial infarction by injection of the stem cells into the injured area. But the question is that if we have senescent senolytic cells, do we need to treat these hearts with senolytics first before we anticipate that the stem cells will give rise to cardiomyocytes? The second potential and very interesting pathway, or the therapy, potential therapy, is to introduce either transcription factors that can be beneficial for cardiomyocytes for their proliferation and differentiation, or block certain mRNAs like in fibroblasts, which prevent the fibrogenic responses. And that will be the reprogramming factors. And usually it can be used either by adenoviruses, like in this case, the authors used adenovirus 8, and that will be the cellular therapy that potentially can be very interesting in the future. In conclusion, cardiac fibrosis fibroblasts are the major source of fibrous scar in cardiac fibrosis. Cardiac fibroblasts originate from endogenous fibroblasts derived from epicardium. Cardiac fibroblasts transiently activate to 7 days. They lose SMA expression during recovery stage. And cardiac fibroblasts that persist in fibrous scar for more than 28 days after myocardial infarction exhibit a senescent phenotype. Senescent fibroblasts do not proliferate but maintain local inflammation and promote senescence of surrounding cells. Senolytics are a new class of drugs that cause apoptosis of senescent cells. Thank you.
Fabulous talk, and it's absolutely remarkable what these discovery science methods can do in terms of giving us insights. We need to work out how we're going to use these. I'm fairly sure that red wine is senolytic, isn't it? I read
Yes.
that somewhere.
I agree with you.
I'm sure we're going to be in need of some senolytics after today.
This is a tough one to be the last lecture.
It is. I have a question for you. This is coming from a cardiologist's background, not an expert in discovery science, sorry. I'm sure you're right that the fibroblast plays a really important role in the recovery of the myocardium after myocardial infarction, but we completely ignore it. And one of the questions I always ask myself when a patient comes in with an acute coronary occlusion and a completed infarct days ago, late presentation, is whether I should open the artery or not. And I guess my question really relates to whether you think that having an active vascular supply to the myocardium during that period of remodeling would be useful for the clearance of these senescent fibroblasts or not, and if you've got any insights from your experimental models that might help us answer that question.
That's a great question. I don't think I can answer this question, but of course, you know, the environment also predicts why this fibroblast becomes senescent, detrimentally senescent, and that is associated with age, with obesity, with many other factors. But what I also want to say that fibroblasts is the only population that I mentioned. So we also talk about immune system, which get tired and get senescence. So of course, you know, like the more oxygen, you know, you can bring to the site, the better the outcome is.
Okay.
So if I give one of these anti-senolytic drugs too early, like in the ER, is there a negative effect, like we're going to blow out the heart?
So at this point, all senolytics are in clinical trials in the phase 1, phase 2, and I don't think that any of these drugs went to phase 3 yet. And nobody really knows how to use them. But I think that the future would be to find the optimal combination of different drugs. Like, for example, one is the antioxidant, and another is targeting a specific cellular population that in combination would be harmless. And probably that would be the solution. But so the person that I also mentioned, Kirkland, from Mayo Clinic, so in one of his lectures he said that so when we think about going to Mars, so that would be not possible without senolytics.
Nicholas?
Because, so when you do myocardial infarction in mice, it's very stereotypical. So it's always the same zone that is infarcted, etc. In human, it's very different because, and so do you think that depending if the fibroblasts come from a zone that is hypoxic versus less hypoxic, it may affect the way they will differentiate into senescence versus the other one?
That's a great question, but I actually think that it's probably more related to like telomere shortening, the RAS production, and again, factors that are more general factors related to this patient. Of course, you know how the zone of infarction affects the fibroblasts and their migration into the zone is important, but I think that the general inflammation in the body would be probably also very important.
And with that, I'm going to invite Dr. De Soma to come up and make the final comments.